Hey everybody, we are going to be finishing up from gene to protein today. So we're going to start with kind of tying up a loose end, and then we're going to move on and we're going to talk about mutations. So this loose end that I'm talking about is signal hypothesis. Way back in the beginning of the year, we talked about ribosomes and that ribosomes are responsible for building proteins. But we talked about two different types of ribosomes. We talked about free ribosomes that are found within the cytoplasm, and then we talked about the ribosomes that are attached to the endoplasmic reticulum. So this signal hypothesis is going to kind of talk about what makes ribosomes attach. And so essentially we think that there's a clue on the growing polypeptide, and that clue is going to cause the ribosome to attach to the endoplasmic reticulum. So what that means is that all ribosomes are going to be the free ribosomes within the cytoplasm unless they are clued in by the polypeptide to attach to the endoplasmic reticulum. So what you're looking at here is a ribosome in the cytoplasm. It is translating an mRNA transcript. You have a signal recognition particle. It recognizes the signal on this polypeptide. It causes the ribosome to attach to the endoplasmic reticulum, making it the rough endoplasmic reticulum. The polypeptide is made, and you can see here that this signal is actually no longer attached to the polypeptide. So now the polypeptide or protein is within the endoplasmic reticulum, and it can be sent to other areas within the cell. So the result is that the protein is made directly into the ER, and then the protein can be secreted once it's complete. And so that clue, what cues in that the ribosome should attach to the ER, is going to be removed during processing. So the first 20 amino acids are going to be removed. Alright, so moving on to mutations. Mutations are going to be changes in the genetic makeup of a cell, and this can be at the chromosomal level or at the DNA level. So for this chapter, we're going to focus on the DNA level, and we're going to be talking about what we call point mutations. These are going to be smaller changes, so we're going to be looking at changes in one or a few nucleotides within the genetic code. Now, it sounds like a really small change, and sometimes it is, and sometimes there's very little effect seen. Other times, though, these changes could be fatal. So we are going to look at a couple different types. We will look at base pair substitutions, and then we will look at insertions and deletions. A base pair substitution is going to be the replacement of one pair of nucleotides by another pair. So here we are looking at regular wild type hemoglobin, then the mRNA that is made, and then you're just getting like a little snippet of the amino acid sequence. Now we are looking at a mutant hemoglobin DNA. You can see that we replaced a thymine with an adenine. That changes the mRNA from an A to a U, and that is going to change the amino acid sequence, and so that's what causes sickle cell hemoglobin, only one base pair substitution. There's a couple different types of substitutions that we want to look at specifically. We'll look at missense, and this is when you have an altered codon, but it still codes for amino acids, it's just not the right amino acid. And then you have nonsense, and this is going to be when you change a codon to a stop codon. So you're not going to continue building the amino acid as you should. You're actually going to stop building it completely because you've changed a regular codon to a stop codon. So again, we are looking at base pair substitution. Here you have no effect on the amino acid sequence, so you have, if you compare this to the wild type, you have a U instead of a C, but everything is still okay. With the missense here, you have an A instead of a G, so an A right here instead of a G up here, so you are going to be changing the amino acid, so it is a missense. Now in this example, it's a nonsense mutation, and so you have a U down here instead of, you can see up here it was an A, and so UAG is a stop codon, and so you've now stopped building this polypeptide pretty early on. So that is a nonsense mutation. Obviously a nonsense mutation towards the beginning of a polypeptide is going to be pretty severe. So a quick question. What will the wobble effect have on a missense substitution? Well, if it's the third base that is changed, the amino acid might still be the same, and so then we say that the mutation is silent. And we say that it's silent because yes, there is a mutation, there is a substitution, however, it's still coding for the same amino acid. So yes, you're going to get the same protein. However, silent mutations may still have a small impact because they could actually slow down how quickly the protein is being made. The reason for that is just that some tRNAs are a little bit harder to find than others. So if you happen to kind of switch out a common tRNA for a less common tRNA, it could actually slow down how quickly the protein is being made. And that's what you're looking at here. So we have an A instead of a G, which makes this a U instead of a C in the mRNA. However, you're still going to get the same amino acid, and so this is considered silent, so there's no effect on the amino acid sequence. Missense effect can be no effect to fatal, and it all depends on where the amino acid was in the protein. So for example, if it's in an active site, that's going to be a major effect. Anytime the active site of a protein is affected, it's going to usually have pretty major impact. However, if this missense it happens in another part of the enzyme, sometimes you might not see any effect at all. With nonsense, this is completely stopping protein synthesis. So a lot of times this is going to lead to non-functional proteins unless that mutation happens like very close to the end of the polypeptide. So if it happens in the beginning or the middle, you're usually going to end up with a protein that is just non-functional because so much of it is missing. And now we have sense mutations, and this is going to be the changing of a stop codon to a reading codon. So this is essentially going to be building longer polypeptides, which may not be functional at all, or may just be less functional, or they function much more slowly. So an example here would be heavy hemoglobin. Alright, now we're going to move on to talking about insertions and deletions, and this is going to be the addition or loss of a base in the DNA. What's really important to remember about insertions and deletions is that it's causing a frame shift. And so that's like when you're looking at these bases, you're looking at them 3 at a time. Well, if you add one in or take one out, you've now changed that reading frame. So every 3 groups, or every group of 3 after that change is going to be affected. So frame shifts can cause like extensive missense, nonsense, or sense mutations. We are looking here at a 1 base pair insertion. So again, we've got our wild type up here. So this is the wild type DNA, mRNA, and then the amino acid sequence. Here you can see we add an extra A into this 3' side, which gives us the extra T on this 5' end. That is going to cause an extra U in the mRNA, and actually by adding it there, that is going to put in a stop codon. The UAA is a stop codon, so we have caused an immediate nonsense mutation with a just 1 base pair insertion. Alright, now we are going to be looking at a 1 base pair deletion. So again, the wild type, so your template DNA, mRNA, and then the amino acid sequence that you would normally make. Here we have a deletion, and so we've taken out the A and T up here. Okay, so this is now missing, which means that the U on the mRNA is missing, and so because that U is missing, you are going to now be regrouping and changing everything after that. So here, instead of having UUU, you're now going to have UUG, and now instead of having GGC, you now have GCU, and this would also most likely affect the stop codon. So now instead of having a stop codon, you've got AA, and then it would continue on down the line. And so you've changed 2 amino acids, you've also removed a stop codon, and so now this polypeptide is going to keep growing. So another quick question. Loss of 3 nucleotides is often not a problem. So why would that be? What do you guys think would cause that? And really it's because if you lose 3 bases, you're essentially taking out 1 whole codon. And so oftentimes you can restore that reading frame, and the protein may still be functional. Alright, and so that is what's happening right here. You are removing TTC, that is pulling out AAG, and so you are completely removing one amino acid. Keep in mind that this happened at a sort of important point because it removed 3 bases that were in the same codon together, and so that's what restored that reading frame. It kept it the same because it removed the amino acids that were all part of the same codon. I'm sorry, it removed the 3 bases that were all part of the same codon. Mutagenesis. This is the process of causing mutations or changing DNA. And so mutagens are going to be materials that cause DNA damages. So we've talked about some of these in class before. We've talked about radiation, so UV light or X-rays we talked about very specifically. But also any chemical that can interact with your DNA can cause mutations. So a spontaneous mutation is also a possibility, and this would be a random error that happens during DNA replication. We talked about the error rate for DNA replication within this unit, and we said that the error rate is really low and that DNA is able to go through, and it's usually repaired after replication, so that the errors are fixed. However, you're talking about billions of bases, and so some errors do happen, and we call those spontaneous mutations when they occur during DNA replication. Just a quick comment, this has to do with like the chemical mutagens. Any material that can chemically bond to DNA or that is similar to the nitrogenous bases will oftentimes be a very strong mutagen because it's able to interact directly or actually mimic pieces of the DNA, so it can oftentimes cause pretty strong changes. All right, the next topic. Now we're going to look at, so what exactly is a gene? We talked about genes versus DNA versus chromosomes. We've used all of these words, but now let's kind of really get in depth about what is a gene. And so a gene is a region of DNA that can be expressed to produce a final functional product. And so the product can be a protein or it can be an RNA molecule. But that gene area of the DNA is going to be a very specific area that can be used to produce some type of final product, and it should be a functional product. So not all of your DNA codes for genes. Only certain sections of your DNA are coded to express a protein or a piece of RNA that's going to have a job to do in the cell. So now, protein versus RNA. A protein is usually a structure or enzyme that has to do with your phenotype, whereas RNA is going to be used more often as a regulatory molecule, and we will talk much more about that in the future. So the protein is going to be something that helps determine phenotype, whereas RNA is going to usually be used as a regulatory molecule. Alright, so here's your overall summary for this chapter. So things from chapter 17 you should know. The Beetle-Tatum experiment, you should be able to explain what we mean when we talk about the central dogma. You definitely need to be able to read the genetic code. So if you're given a sequence of DNA, you need to be able to transcribe that into mRNA, and then take that mRNA and then translate it into the amino acid sequence. You need to be able to describe the events of transcription and translation, what happens at each step through those processes, what enzymes are involved. You should be able to talk about RNA and protein processing. And then you need to be able to talk about mutations and also what is a gene. Alright, so your lecture question for today. So can you guys explain to me what kind of impact a frame shift mutation would have at the beginning of an mRNA transcript compared to a frame shift mutation at the end of the transcript? Alright, so explain the difference between a frame shift at the beginning of an mRNA transcript compared to the end of the mRNA transcript. So please send me a message with your answer to that question. Thanks, guys.